efforts we've had was trying to get some octocorals and to get a slit shell, because these, we just don't know what these are. And unfortunately, we have already collected pretty close to what we're allowed to collect. We're the policy is that, yeah, we're at the limit. The policy is we can collect 4 samples, and if you've been keeping track, we've collected 5 today. So I'm
Close
just
to limit, huh?
Yeah, we are over the limit, so I'm not sure what to do here. This is the dilemma that we face. Do you collect right in the beginning of the dive because you may not see it later on? And if you collect all these things towards, we've got virtually an 8-hour dive. Wait till the end to collect. You may have missed something in the beginning. I can tell you for sure that our coral people would have loved to have collected that pinkish colored octocoral that we just saw, and I can tell you without a doubt that this is a high priority for collection as well, so I'm not sure what to do. Let's go in and get some closer imagery on that, please. Yes, please.
Push closer.
Now we're at 285 meters. What were the
Your mic is not open.
The depth range at which we saw this in the past?
Mic's not open.
Hold on, let me look at my notes.
Open your mic.
Yeah, she doesn't.
So we're wondering about what the depth range was?
Shirley, I don't know if Casey is there right now. She's more the consultant for whether or not we can do a one-time breaking of the rule here. This gastropod, of course, generated a lot of excitement by a bunch of people, and of course we weren't expecting to see it. We saw one way down further as we're transecting up, but I hadn't really thought about it before. But, if Casey was willing to make an exception here, I would support the decision to go for an extra one.
Well, I will see if I can find Casey on the ship. I've sent her some email earlier in the day and didn't get a response, but I'll step out and see if I can.
Hey, come on out, video. Gonna get closer.
They're going to go and make an announcement.
Pilot, are you just going to hang out here until...
Yeah, I'm going to hang out a bit. Yep.
We saw a slit shell 3 days ago at 334 meters.
Of course, I have a current now that's just spinning me around.
So I'm just wondering if they kind of like this depth.
They occur at different depths. Different species occur at different depths.
Okay, because we'll be on Ifuku seamount the day after tomorrow in a 350, 5550 depth range. Do you think it might be possible that we would see one there?
Don't know.
It's just
You're muted, Patty.
Go ahead and roll.
We are going to be diving on Ifuku seamount day after tomorrow, and that's a 550 to 350 meter range, and the same depth range on Daikoku seamount the day after that. So just wondering, folks, if you think that this is a similar depth range for seeing this individual.
Pilot, co-pilot, what do you think about changing our heading?
Well, the issue is, how active those other sites are. These are geology guys, and they were selected for, they were hoping to find pretty recent active volcanism. And I don't know, for this shell, maybe it doesn't like environments that are too active. So.
Understood.
Yeah.
Here,
go ahead and make that
we know that
heading
there's a lot of
change.
interest in it. That's all I'm saying. Maybe we could make a rare exception.
Well, we have plenty of time left in this dive, and we can sit here a bit, take a look at it, and wait for consultation with our expedition
Watch lead pilot
coordinator.
Okay, sure. I'll let you know when we're done with this sample. Thank you.
Go ahead, Watchley.
Yeah, we're okay sitting here for a while?
Sure.
Okay.
Understood. I'm watching this one. we're just going to hold here for now.
Great, thanks. Yeah, it could
You know
crack
what that
for
was?
sure.
I think it's me.
I think it is.
Is this? Yeah, fix the ground on that.
Pilot, you want to stripe it with lasers? Could your claws grab that?
While we're
don't know.
waiting here, nope, that's not Casey. While we're waiting here, I was just wondering if any of the biologists on shore
Pilot,
have any
we'll add
these
that to
about
the,
these
electrical
holes that
test.
I'm seeing in
Yep.
the ash layers. And what I'm wondering is whether
We can do that.
if ash is deposited on the seafloor, it is going to be unconsolidated, perhaps, and would animals like to use this as a medium to burrow into to escape predation? Because we do see pockmarks on just the top layers of this ash in place. We don't see it so much right here on some of the blocks that are broken off the outcrop.
So, if we decide to sample this, we're probably going to want to go. Oh, the easy boxes are filled.
They are, and we have specimens we don't want to float out.
What's in the
both
port
ports
side?
started.
Patty, I'm not sure if you were referring
Side,
to
we
the
have,
unconsolidated ash,
Yes.
like in a
Geosample
sediment
in
form,
inner,
or
and we also have a piece of
this is
coral
once
in
it's
inner. We have nothing
formed
in
into this
port
rock-like form?
No, I
starboard
was
inner
thinking
is,
of,
that's heavily
shortly after it
displaced
has
air, and starboard outer
been
is empty.
deposited and before it,
Algers.
Both
I mean,
outers
some
are-
of it may be
think I'm
welded, but
gonna
some
have
of
to
it
go
may be relatively
outer.
loose and then eventually be cemented
have to wing
essentially
in.
by alteration. But during the period
On
after
the,
it's deposited and
port
before
side
it
there?
is,
Yeah.
starts getting
So actually, we
lithified,
can do that now while we're waiting.
I'm guessing
Let's,
that things could burrow
retract
into
the
it,
pins.
and just wondered what you thought, what kind of animals might
Port
do that.
retracted. Starboard retracted.
Bring the port wing in.
Sure,
inboard.
I guess I don't know why burrowing animals wouldn't use it. There are different reasons that some of these invertebrates burrow. If they're trying to get food, then they're probably not going to be burrowing to fresh ash because you wouldn't have enough organics mixed in with it. But
Yeah,
there are
let's see if
other
you can extend
various
the draw.
crustaceans, crabs and shrimp and worms and
Both
so on that will
retracted.
essentially make a burrow as a hiding place to hide out from predators and then emerge from it to feed. And
Nice and slow.
the question would be, what did the bottom look like here prior to the deposition of ash? And are those burrowing organisms present to take advantage of the ash? And if they aren't, how far away do they have to come from? So it's kind of a recruitment timing issue, I would say.
Yeah, I suppose.
Yeah, I think we're good.
Yeah, there should be plenty. Yeah.
Another question would be,
Well,
how fine a
yeah.
grain is that
Yeah.
ash? If it's too fine
And then,
or too coarse, that is going to
fire
affect
up the
what kind of
shilling
species
and
can use it, and does the
move it
burrow
to the
have
port
to
a little
be
bit.
lined with some mucus to stabilize it, or a tube,
Yes, sir.
or is it fairly coarse that's not an issue?
Go ahead, sir. Damon.
So there's all kinds
Port
of
back
different
like a neighbor.
parameters that would go into determining what sort of fauna would
Yes,
create
we
a burrow
have
if the
five samples.
substrate were
We
available.
have,
Oh, that's
ceiling
pretty
pools
interesting.
tomorrow.
Thank you.
What would you like me to do with the shilling here?
Can
So,
you bring it out to
we
port
have
anymore?
That's your call.
Casey
We can do
Cantwell
it if
in
you
the
want, and we do have
lab
space
now,
for
the ROV control room.
Actually, I
And we're
think we're
We're not
okay.
gonna collect
Yeah, I think
it.
it'll
we
be
are
good.
discussing this issue.
Okay. Pilot, we're not going to be able to collect that sample.
Okay.
Hi, folks. This is
Bring
Casey
the board
Cantwell.
outboard.
So, as this is our would be our 5th or 6th sample for the day, we cannot collect it. I know that we were hoping to get one of these, but we do have another shallow dive at Chamorro Ridge. It's a little bit deeper, but still shallow, so it's probably within the range for these guys. And then
Valpex
we
is
have a dive at
right
Explorer
now.
Ridge about 7 or 8 days from now, where we also will be in a similar range where we could potentially see this again.
Don, set in.
Sure.
Are there any questions? I know we've been trying to get this one, so I apologize. All right, thank you very much, guys.
Thank you, Casey.
Watch lead nav.
Go ahead, watch lead.
Yeah, how far away from our next waypoint?
Our dive end waypoint is about 80 meters south of us.
Hi, Don.
although we haven't been following
Hello.
that
Good
path
afternoon.
exactly because the ridge has been
Good
a bit,
afternoon.
to the west. So
Welcome
80
aboard,
meters
pilot.
is the answer.
Okay, thanks. Okay, what do we want to do here?
Explore the seafloor.
Okay, so we are going to continue exploring the waypoint. Our end waypoint was this about 80 meters south of here?
Yep.
Okay.
14.
One of the things we had discussed doing with the group that's on the chat was to perhaps take a look at the steep slope on the west side of the scarp.
Yeah, we did talk about that and seeing if we could actually just drop down a little bit and then move
then move up again.
Yeah, like
and
maybe
do a
just
vertical
about 30.
transect.
Yeah, I think we were talking about that yesterday. Maybe move down about 30 meters and then come up.
Right.
Yeah.
right.
Yeah.
It might give us a look at different habitats.
Okay, so where are we in relation to that?
Oh.
Yeah, watch lead nav. Did you catch that?
Sorry, watch lead. This is now. What was that?
Yeah, we were just talking about going over kind of to the edge.
Starboard?
Yes, yeah, to the edge of the escarpment, dropping down, and then working our way. Well, either dropping down, going down a little bit. I know you don't like to work downhill.
But just dropping off.
Just dropping off and then working back
dropping
up.
off into the water column.
Okay. Did you want to do that to the west of the ridge?
We want to do that on the steepest side. So in the nav screen that I'm seeing, it's if north is up, then it's to the east.
Yeah,
south
to the
and
southeast. Yeah, southeast.
South and east. Okay. Stand by. Let me talk to my pilot about that. Pilot, they'd like to go southeast down off the edge of this ridge.
So they want to go down and then come back up it? Is that the idea?
Yes, that's right.
Down
Okay.
and then back
That's
up.
right, pilot.
Yes.
Yeah, we can do that.
Thank you.
So Don, let's stow my upper swing arms.
And watch lead, we do have just...
Roger, I'll stow the upper swing arms.
Thank you. Thanks very much.
Yep, and then it's upper, which is the lower right. Upper swing arm, stow. The uppers you can, it's impossible for them to hit anything. So the order you do it doesn't matter, but you need to stow and then you need to tilt up. Just make sure you're doing the uppers, that's all.
I'm going to call the bridge
Upper
and
swing
let them know they can
arm,
make that
Yes, so for those of you who didn't hear the conversation, what we decided, because we were talking about this, I think last night when we were doing our planning meeting, that if we got far enough along in the in our
Ken, go ahead and make that heading
track,
direction for-
that had enough time towards the end of the dive that we would do a kind of drop off the escarpment and work our way up. So that's actually what we are going to do. So it's a little bit southeast of where we are right now. We're going to drop over the edge and then work our way back up.
Pilot, are we ready to make that move to the
What's our bearing on the move?
really could be,
Okay.
let's say, 130.
130.
That's based on the multi-beat,
down.
though, which seems a bit off here.
Well, I just need you to tell me what our bearing is going to be.
Yeah, 130.
130. All right. So Don, we're going to come to 130 here.
Roger, copy 130, and the hydraulics are down now.
Okay, thank you.
And this is going to be a pretty significant run.
I'm gonna turn to the left
maybe 100
to
meters.
130.
Yep, that's fine.
That'll put us at my cursor on high pack.
So Don, if you could set your camera at about 45 degrees.
Roger, copy. Set it for 45.
All right, and then we're gonna let's make
So
that move.
Jeff
Let's do it
Drazen
pretty
is
quick.
in the ECC now, and he
4.3.
sort of pointed out an interesting fact that
Then
on
we're gonna
his
pull up a
dive, of
little
course,
bit,
he's
Don.
a geologist, and on his dive, the
Roger
Hadel
that.
Ridge dive, we virtually
by
saw
on that.
no animals whatsoever, but it turned out to be a marvelous
Is it
geology
possible for you
dive.
to make the ship move
And
while
on this particular dive, Bob Stern, a geologist, picked this site, and it turned out to be a marvelous biology dive. So I'm wondering whether the geologists ought to pick the biology sites and the biologists pick the geology sites.
That's it. We could think about that. We could certainly think about that. But you know, Chris, you did point out that one of the things we were going to be doing for this dive as well was to look for like precious coral and
fish.
and bottom fish.
And
So
we saw them.
so we certainly did see that.
So let's do point 2, and we'll stay in contact with the
This,
bottom.
this is better than any of my three dives, so clearly Bob getting the fishery stuff here.
fine with doing that downhill?
Oh,
Yeah.
that's
How
good.
long? will
And
that
look
move
at all
take?
these barnacles just edging
The 100-meter
the sides of these boulders. That's just so strange.
What's amazing to me is that each one of these areas where we've been diving has been different. Every single one.
yep.
I mean, even the ones that are like within the same depth range, all different.
Well, I
A
think
lot has to do. Right. Go ahead.
I think Mary Jo, was it?
Okay,
I'm sorry
I'll
if
call
I
it in now.
misquote whoever said this, but I think maybe it was one of our pilots said that the substrate
we
made
select
the
different.
difference.
Absolutely.
And
Pretty
it's
much
true,
forward
and there's a
for the ship, so
grouper
that,
down there.
should be
It's
fine.
true that the material, the dark material that we're looking at right now with all the barnacles on it,
100
it
meters.
is coarser grained. That means it has
130
larger fragments
0.2
of
knots.
volcaniclastics, actually two groupers. So that might make the surface. I'm waiting to see if they come out the other side.
copy,
I'm
thank
wondering if that block that we're looking at right in the middle of the screen
It's just a slab.
kind of makes a little tent. Yeah, just fell off an outcrop.
Where's that going to put us?
Oh, nope, they're not
I'm
coming
about
out.
to drop a marker there. Well,
Anyway,
that'll
I
be
think they're
finished.
sticking onto the material.
There-
that
yes, it
has
did.
coarser. They did. Okay.
Brighter.
It's a little tunnel for them.
That's the ship. And actually, you're pretty much under the ship now, so it'll be.
Those
right
are
on my
big
cursor.
groupers.
They are
Yeah,
big
no, that's
groupers.
it. Lost them there. There it is.
Co-pilot, we should be
This
starting
darker
to move,
stuff is
just
not only
about
coarser
now.
grained,
The,
but
Roger that.
has
Copy.
thicker
130, you
layering.
said? And we're heading downhill.
Perfect.
Nice swimming.
So we have just about an hour left to go on our dive. So we're going to finish it off really nicely by
The ballet of the
driving
groupers.
this, dropping down and moving up the slope. Currently at 286 meters.
Pilot, you happy with that camera angle there? You want me to just leave it?
Yeah, that's fine.
Okay.
And
There are 3
you can just
groupers in
leave your
view
mic
now.
where it is. You sound pretty good.
See the tail of one
Yeah.
under the slab of rock, and 1 each on the entrance and the exit side. Four. There were 2 under that slab. Are groupers territorial at all?
I don't know.
Bruce, you want to handle that one?
Well, I don't know, but they're often seen in these aggregations, like I mentioned before. So it seems like they're probably not, at least at the times when they form these groups.
Thank you, Bruce.
Chris, do you have other thoughts about that?
Right. So protogynous sex change or protogynous hermaphroditism is often associated with a haremic social system, which means that the male, the dominant male, if you will, the bigger individual, will actually have a territory, and there will be 2, 3 to 4 females that are with him in that territory. And so something like that may be going on. So even though you see a grouping of them, a bunch of them together that are getting along well, the males actually may be excluding other males and other females from their territories.
Come in on this.
Okay, understood. And we have an anemone. This one is open.
Yeah, pretty, like the ones we've seen.
Beautiful.
Very pretty.
Maybe not fully open.
So this one is semi-closed up
Yeah.
completely.
That's what I was thinking too, Chris.
In her mouth.
Really pretty. Now, is that coral another Parasus, or is that something different?
There are 2 different corals down there
Yeah.
right here. The one on the left is obviously different.
Those look like 2 small colonies of Parasus, or what we think is the genus Parasus.
Do you think they're both the same genus?
Yeah, I do. I think on one of them we're seeing more
polyps
out,
extended,
so
yep.
you don't
Right.
see the white as well as you do on the other one.
Right. Okay.
About 75 more meters in this move. The course we chose seems to be a good one based on the sonars. Might be a
This
little
slope
more
looks as if
starboard.
it's made up of an awful lot of cinder-sized chunks of broken up volcaniclastics. Looks like it would be a really difficult type of slope to hike up. Very slippery. We seem to have a lot of the same sort of organisms that we saw very
Like a
far up on the top.
Yep. A lot of fish in the water. Seeing some crinoids. What is that? Oh, more grouper in the distance. Urchins.
Come in on this.
Some of your encrusting sponges.
Yeah, some of the encrusting
Big old
sponges.
grouper
Yeah.
down there.
Come on up. What's the depth at the bottom?
Fish is back.
The part of this seamount that we are descending is on the east
310 to 320.
of the shallowest point that we reached during the dive. On a map view, that would be, we're going down sort of the center of a concave or arc-shaped, I guess I would describe it as a cut in the flank of the cone that formed on the seafloor right here. It looks as if the southeast half of the seamount may have broken free and slid down the flank of the edifice toward the southeast. So basically we have an arcuate scarp
Keep going here,
that
Don.
we're descending, and
start going down
we're
slope.
looking
It's going to get
toward
a little
the
annoying.
chunk of
So
material
you're
that
gonna
may have slid down the
want to keep
flank.
an eye on your altitude.
But we'll turn around eventually and face sort of northwest and come back up the slope, I
Yeah,
think.
co-pilot, it looks like the current is pulling your tether off to port, which is a great direction. There's open water there. On starboard, there's quite a bit of rock.
Okay, roger that.
Partial.
Halfway done with our move, 50 meters left.
Those lasers are 10 centimeters apart, so you can get some idea of the scale, the size of the things that we're seeing here.
Hey, come on out.
I'm running plus 6 to be gain.
Yeah, now I'm looking out into the abyss.
Yep.
We wish.
That's the direction we're headed.
Yep, that's. So, Scott, could you explain the difference between an octocoral and what most of our viewers would know as stony coral?
Sure, I'd be happy to. So all the corals belong to the phylum Cnidaria,
What was the
which is
plan on this
basically the
meeting? Trying
group
to
that
get to the
includes
bottom
the
again.
gelatinous
we're gonna
animals
yeah, exactly.
that have stinging cells called nematocysts. So if you basically, if you're an animal that possesses a nematocyst, that is, you produce them yourself, you basically belong to that phylum. So that includes all the jellyfish and the sea anemones. Corals are cnidarians that have
They had
a
a target
skeleton,
depth
or
for the bottom
typically have a
of the-
skeleton of some kind.
It'll be about,
And there are several different
310.
groups that can be referred to as
Yeah,
coral.
well,
And so the most common one, the one I think that most people are familiar with,
What's the layback
is
at
the
this step?
scleractinian corals, or the
Oh, it's
stony corals, and those are the ones that produce
Have to
things
look at the
like
yellow
Australia's
coating on
Great
the rock.
Barrier Reef and many of the tropical reefs
Maybe
that
10 meters.
people love to scuba dive and snorkel on. Create a lot of structure, along with sponges. I got to point out the sponges that keep those reefs together for Shirley. And essentially, when you look at a stony coral, it creates a skeleton that's calcium carbonate, and the tissue is sort of draped on top of it. So you can think of the underside of the tissue as depositing that skeleton as it grows. The octocorals are a completely different group. They're not closely related. In fact, sea anemones and zoanthids are more closely related to the stony corals than octocorals are to the stony corals. They're in a separate subclass. And octocorals have an internal skeleton, and mostly that skeleton is made of a protein, and it's somewhat flexible. So if you think of something like a sea fan, many people have seen sea fans when they're snorkeling. You can see that it's flexible. It may move back and forth in the current, and we've been seeing that on many of the octocorals that we've seen here. That's what your typical octocoral would look like. Most of the stony corals and most of the octocorals, almost all of the octocorals, are colonial, which means from a single egg, hundreds to thousands of polyps will develop, all remain connected together in a common tissue and have that skeleton on the inside. So each of them are feeding on their own, each of those polyps. So those are the basics, but there's a lot of variation in there. So there are some some octocorals that do have a more rigid, stony, calcium carbonate-like skeleton, like the bamboo corals that I'm most interested in. The primnoids that we've been seeing here have protein with calcium carbonate embedded, so their skeleton is a little bit harder. There are many of these stony corals that are not colonial, so we've been seeing what we've been calling cup corals and so on. So there's a lot of variation. We also talked about black corals. Black corals are actually more closely related to the scleractinian corals, the stony corals, except that they also produce a protein-like skeleton.
You got to keep an eye on
but
my
as
tether.
the name implies, that skeleton is black. It's also thorny or spiny, sometimes called the thorny corals.
We just don't
and
want to
those are
snag it
one
on
of the
anything.
precious corals. So the
It's
black coral
unlikely to happen,
skeletons
but
are used to
if
make
that
jewelry.
happens, it can be.
And then finally, there's the hydrocorals, which belong to yet another group. They belong to the hydrozoa,
It looks like
which
they're
is
well
what
up off the bottom.
includes the siphonophores,
You get
the
the nice
Portuguese
footballs,
man of war, those
so.
sorts of things that we've been seeing.
All you need.
And we've been
worst
seeing
case is
some
you
things
like hook it
called
under a rock,
stylasterids
knock
that
the
we've been
rock
referring
loose,
to.
and now
Those
you
are
have like a
hydrocorals.
5-ton drop
Those
on
also
your
have a
tether.
very stony skeleton,
Anchors
but
are not
typically in the
good.
shape of a fan. So that's the sort of back of the envelope introduction.
Excellent. Thank you very much, Scott. What are we seeing with the geology here, Patty?
We're seeing a whole bunch of really large boulders of essentially the same sort of stuff we saw at the summit. And as I said, looks like we are descending down a slump scar that is just cluttered with large chunks of the material from the top of the volcano that have rolled down this incredibly steep slope.
Fewer fish down here.
I have a feeling that this material that we're looking at right now makes up the bulk of the edifice, at least at this portion of the depth range that we're looking at. So I think that the samples that we've recovered are going to be highly representative of the major type of volcanic product that this volcano has erupted. So Shirley, is this sponge material that we're seeing on the seafloor?
Well, it would be nice to you know, this might be that white sponge that we saw
That's what I'm thinking.
earlier. We can
Just
take
encrusting stuff.
Yeah.
Impossible to sample.
Yeah, watch lead pilot.
Go ahead.
Yeah, can we just take a closer look at that white sponge? I see him looking at you. I'm watching you guys from back here.
Yeah, we'll be able to. I'm just gonna wait until co-pilot's a little further out.
Okay, yeah,
And
because
then,
her rear end is kind of perhaps close to the
We have
slope.
about 10 meters left, and it should move to them. We'll be able to take a look.
So I wonder if a couple of those stock things that are coming up that are not the crinoids that seem to have some squat lobsters or something on them. I wonder if we could get a zoom on one of those.
The tall ones, you mean, Chris?
Not too tall, but tall.
Yeah,
he's
relatively
got one
tall.
right in the center of the screen right now. It's exactly what I was talking about.
Looks like it has white specks on it.
Yeah. So we can do that right after we look at the sponge.
And there's a shrimp swimming by. And a fish.
Certainly has been a really interesting
Very biologic.
Yeah.
fascinating
Yeah,
biologic
lots of stuff.
dive.
Lots of stuff. Too much stuff. I
Oh,
wanted
yeah,
one of
I know.
each. I wanted
Go
one
down
of
and
each.
look at me, Don.
Don't we all?
Roger.
I know.
Because you're gonna sort of come up over on top of me. Got a rock right behind me.
As we mentioned before, these samples are available for anyone who wants to study them. So they're not my samples. They're not.
Not my samples.
No,
that's good.
not Chris's samples, not Scott's, not anybody's. They're everybody's. So anyone who wants to study these samples or look at the data,
Yeah, you
look
can
at the images.
request
Yep.
the data
You
and
can-
find all of this video online at the Okeanos Explorer website, which is absolutely great. I mean, you don't have to sit through the entire dive. You
Oh,
know,
yeah,
you
there's
can,
all kinds of, yeah.
You can look
Come
at
down
some
a
of the
little bit.
clips
Coming
that our videographers are
3
getting
meters.
as we're diving. They're cutting out little bits of the video and incorporating it with some of the descriptions that people on shore or Shirley or I are making here.
Yeah,
So
and yeah.
most of these images will have a little description of what it is you're looking at. At least that's what we hope.
Yeah, and the Office of Ocean Exploration and Research at NOAA is actually, they're developing more and more tools. Just about, I think, it's ongoing
Yeah,
development
absolutely.
of tools that it will enable us to filter out the data, search on specific types of data that we're interested in. So the data are going to be available, I think, within 60 to 90 days. I don't even think you have to specially request that. You have to be registered, I think,
Right.
as a scientific user, but don't have
How's
to.
that?
It's nothing where you have to make a request to borrow it. It is freely, publicly
no,
available.
78.
Oh, keep
And you
coming
can
down.
search
okay.
right,
using
so what we're gonna
metadata
do
such
is,
as latitude, longitude, depth
Yeah.
ranges if you're interested in that, rock types. I mean,
I want to turn around.
anything that
Okay. And start working
suits
back
your
up.
fancy.
We are
Yeah.
about finished with
And
that.
then the samples that'll be deposited
say,
at
Dan?
the
We're
Smithsonian
about finished with that.
And
Roger
rock
that.
samples
At Oregon State.
All right.
and the
do
molecular
the same.
samples at the Ocean Genome Legacy at Northeastern University. So each of these sample sets will will be available to study. So our ROVs, it's a two-vehicle system. So the Deep Discoverer is attached with about a 30-meter tether to the Sirius ROV, which is attached with a much longer tether to the ship, the Okeanos Explorer. See, now this bottom type looks a little bit different to me. It looks more like rugose, more rough.
Well, it's rubbly.
Is that what it is?
Rugose generally means more like corrugated.
Oh, okay. See,
Yeah.
with sponges, we actually use corrugated as like look
Okay.
at the sponge surfaces, and then rugose is kind of
Rough
like this.
regular?
Yeah,
Oh, okay.
yeah. Okay. Wow.
Now
Wow.
that is a
That's a
sponge.
sponge. It is a sponge. You can see the canals of the sponge. You can see like little lines. There
Little
are
dark
things.
lines.
And so you see the epibionts and sediment on there, but the areas that are clear are usually the areas into which water is either, water is being pumped out. So it kind of makes that, washes
the
that area
surface
away.
off, right?
Yes, yeah.
Got it.
Thinly encrusting. These things are probably about, I don't know, millimeter or two thick. Really, really thin.
And the reddish brown material you see on the upper
Oh, now
right
for
of
full
the
size.
screen
No, what's that guy in the
is
top right?
what we call volcaniclastic.
Clear thing.
I guess I would call these lapilli based on the size. That's just a size description.
Yeah, what is that thing at 1 o'clock?
The thing
see?
that's
Yeah,
the
what
white
is that?
hair.
What is that? I don't know. What is that?
Get any closer, pilot?
Yeah,
I
really
have to come
clear.
out.
Sort of translucent brown
Yeah,
thing.
yeah.
Okay.
I'm gonna get closer.
Jeff Drazen says tunicate. Sea squirt.
tilt your camera down and get it right between your...
And do we have
Skids.
barnacles on that block at the top of the screen?
They're
awful
yeah,
small.
yeah.
I don't think you made much.
So, okay, we're going to go back in and see if we can get a closer look at that.
Tunicate. I want to see it.
Yeah,
Oh,
yeah.
that is
That's nice. It's
amazing.
really nice.
So the or I should say oval white
It's one of the
opening,
siphons, yeah.
that is either an intake or an outflow valve
Yep.
or opening, I should say, for this animal.
Siphons, yeah. What's got here?
I'm trying to figure out where the other opening is. Usually there are two.
It's the translucent, and it's
one of the openings is
pointing up at a
ringed
1.
with the white, and then the other opening is just behind it at 1 o'clock.
Yeah.
Not ringed in white.
Yeah, I think I see what you're talking about. It's a little bit harder for us, for Shirley and me on the ship, because the large screen is about 7 or so feet away from us in front
At
of
least.
the pilots. And so we're standing up sometimes, leaning over there, trying to get a good look. But our videographer has a really good high-def screen. Often he will identify things in view before we see them ourselves.
While we're looking at this, I think it's worth noting that tunicate is the invertebrate that is the closest relatives of vertebrates, including humans.
Stars.
That's right. What makes it like that? Is it a chordate?
Yes. So I'm sorry, were you asking that?
Yes.
This is the back
We
side.
were prompting
We
you.
were prompting you. I know the answer.
Oh, I'm sorry.
No, she doesn't. No, she doesn't.
Shirley.
Let's take a look at that coral. Yep. So it has a notochord
Turn off watch too.
Well, at
and
least
a
the
larval
larval
form.
stage
The larval
does.
stage
These
does.
are
Correct.
larvae of that. I'm sorry.
Yeah, go ahead, Scott.
I was just going to say, if you saw
Just
the
turn off
larval
watch two and
stage
listen.
of
Okay,
that organism that we were just looking at,
And
that
watch
transparent tunicate, it would remind you a lot of a little tadpole, or the tunicate. It would remind you a lot of a little tadpole, or there's some small fish called Amphioxus. It looks a little bit like that, and it appears to have a mouth at one end, and it has a long tail. It's got musculature in the tail, and it has a notochord, which is a stiff cord that gives it support.
Yeah,
It's
come
got
on
the
in.
nerve cord in the same place that we have our nerve cord on the dorsal side, and so all those characteristics show that it's closely related to the vertebrates.
Okay, coral specialists.
And now we're seeing another primnoid, Calogorgia. And it looks like we've got some brittle, looks like brittle stars on there. Yeah, brittle stars. Actually, it looks like there
Looks
are a couple
like we
different
have
kinds.
two different kinds yet.
Say that again.
It's not on.
It's not on.
These look like little octopus almost. The two
Turn
on
off
the
here
end.
and turn off listen and talk.
I don't have anything on. I'll turn it off completely.
Right.
Is that any better?
So both Tasako and I believe this is a primnoid coral in the genus Calogorgia, and it has alternating branching. So I don't know how many Calogorgia have alternating branching. The one in Hawaii that's alternate is Calogorgia gilberti. So perhaps it's that, or perhaps they've got a different species here. In Calogorgia, there are other ones that have opposite branching. And what I'm talking about is if you look along one of the main branches, you'll see these branchlets coming out of it.
and the branchlets are coming out, one on one side, and then it goes up a little bit, and another one on the other side, and then they're sort of stair-stepping up. When it's opposite branching, the branches come out of that stem at exactly the same place opposite each other. And so the opposite branching one in Hawaii is Calagorgia formosa. And so my best guess, this looks alternate to me. It's a little confusing here, but Calagorgia for sure.
Yeah, but we're fine.
Well, not for sure, but best guess.
Yeah, watch lead pilot.
Go ahead.
Yeah, could you, we'd like to get a closer look at the things that are a little, you're focusing in, Roland's focusing in on it now. The ones that are not obviously looking like brittle stars are more thorny.
This guy on the left?
Yeah.
I think I was wrong, Shirley. I believe they are.
Yeah.
The upper side of the arm, and I thought I was looking at the underside of the arm.
Yeah, I know, Scott. I was looking at that too. It looked like it was too thorny for a brittle star, but it's just a different kind.
Sure looks like a brittle star.
Yes, that was it. Thank you.
Incidentally, I think all of the polyps here are closed, if I'm not mistaken. And when they close up with Calagorgia, they have a really distinctive shape. It's almost like a little short worm that's cooling down or coiling up or down toward the stem. So that's just another clue that Andrea Quattrini has also chimed in that she's pretty sure this is Calagorgia. And there's a stem that's got some polyps extended now, so you see the difference between the closed and the open configuration, if you will.
So, pilot, the bridge would like to turn the ship a bit to port again, if we can.
I have a poll for the scientists. This is Roland, the video engineer.
Or should we do that
Each
now?
of these polyps, now are each of these polyps one animal, or are they just part of the colony of the total coral, and the entire coral is considered a single animal?
The coral itself is considered one animal, and each of the polyps is a unit making up the entire colony. And so this whole fan-like structure that you're seeing
when they rotate the ship
arose
like
from
that, where is
a
it
single
rotating?
fertilized egg, which
Stern?
then developed into a larva, which swam around for some period of time,
So it
or
shouldn't
crawled
affect
around,
us too
and then
much when we
started
do
to
this.
bud off new polyps, and they all remain together and create a skeleton. And Chris nicely noted there that we have some of those polyps that had closed up and retracted, just like the anemones we saw earlier that had their tentacles curled in. And then some of the polyps were opened up and looked a little bit more fuzzy. And one of the things that we use to try to identify these primnoid corals on the video is when the polyps close up, do they curl upwards and toward the branch, or do they curl downwards and toward the branch, or do they just stand out perpendicular? And amazingly enough, that has some taxonomic value.
Talk about.
And we are zooming in, I hope, very shortly on a green-looking something or other. There are two of them in the field of view. I don't know whether that's biologic or geologic.
Right, come on in.
See now that, yeah. So we're definitely seeing small sponges. Those little cream-colored knobs are sponges. Yeah, different, kind of irregularly shaped, but small demo sponges. That green, I don't know what that is. Boy, I don't know.
Pilot, you want me to swing a little bit so we keep you and tether in view there?
Yeah, I mean, if you.
To me, it doesn't look like a bacterial film, so I don't know.
Watch lead nav.
Yeah, nav.
Yeah, we have
Barnacles
just
on the
about
top corner.
I believe 30 more minutes on the bottom. Did you want to
Go up.
try to head back to the west up the slope?
Yes, please.
Okay, I'll get
Come on
us,
out, video.
get us moving.
Get out.
You know, I'm starting to think these little flat things could be tunicates. I think we saw them the other day.
What? Which little flat things? The ones that were on that rock?
Yeah, I could see on some of them that looked like they had two openings.
Yeah, no, they're sponges. They're, you know what, Chris? I think they're geodeids. There's some geodeids, like maybe Caminus or something like that.
Back to the west.
Upslope.
Upslope.
Give me the flat white things
Yeah.
that kind of look like limpets. So you're saying they're the, okay, sponge, got it.
Yeah. The ones that have kind of, some of them have a couple of like raised oscular areas. They're irregular. They're sponges, Chris. Probably geodeids.
Got it, yep, okay.
Okay, come on in, video.
Wow. There's a thinly encrusting demo sponge with an octocoral on it, on the
Give me
base, occupying that same space.
Sorry, I didn't have my mic open yesterday.
Oh, wow, the polyps are really beautiful.
My iris and black levels adjusted.
Any IDs on this?
And is there a barnacle or two of brown things on the base?
On
the lower
I
part of
see.
the-
It looks like it's a
Oh,
hermit
it's a
crab.
hermit crab. Yeah, you're right.
Sorry, I was exchanging some comments with Andrea Quattrini. So this is an octocoral, and I don't think it's one we've seen yet. And I'm not certain, actually, even what family it belongs to.
Oh, wow, really?
Because it's in one of the groups. And I'm just not too familiar with these groups at these depths. But it could be a gorgoniid, or I'm not sure if it could be an Ellicelidae, but let me think about this a little bit.
Asako is thinking it might be a plexaurid. Something we collected that was Euplexaura.
Going for full
Yeah, that's
specimen.
certainly another possibility. I thought that the polyps looked like they retract too much to be a plexaurid, but it could be.
Mm-hmm.
Good with lasers. Video's clear.
There.
We have about 25 minutes left
Okay, pilot,
in
so,
our dive.
Move at 270. It looks like that'll
Move
be directly
heading
up the
back
slope.
up to the up the slope here.
More like 250. That'll be toward our
There's some
dive
nice
waypoint.
layering in this volcaniclastic material,
Any preference?
and
you gotta
a few
stretch out
really
first,
big boulders
Okay.
rolling
They're
down the
making
hill.
noise for a little bit.
Well, they
Yeah,
rolled
they-
down
they
the hill
rolled-
sometime long ago. Well, I
Maybe
think
from
in
this
geologic
distance,
terms.
If you were a little bit down, shoot across that rock to show all those fish.
Okay, we
above it.
see some more of the fan-shaped octocorals at the top here. Lots of stuff in the water. They have plenty to eat, that's for sure.
Come on in, Rowan. Come on in.
There's a whole bunch of little fish right above the top of this giant boulder. And here's one of your sponges.
Maybe, but maybe bacterial. That
Really?
may actually be bacterial.
Maxim.
Nah.
It looks to me like bacterial mat. All those little knobby things are sponges. For
Oh,
sure.
yeah. It's amazing how much life there
It
is
is a lot.
on a square foot of seafloor.
Yeah, come out
I
partial.
know that sponge back there. Not sure if that's hexactinellid or, ooh boy, or a demo sponge.
I'm gonna come back in on that sponge. Get some more light on it.
What do you think? Oops, oh, who pushed me on speaker? Somebody's on speaker.
I think
There you
this
go.
looks
You
more
pushed it on
like
speaker.
possible hexactinellid. What do you think, Shirley?
Yeah, I think hexactinellid.
Pilot, you're kind of wrapping around this rock. I think you're aware of that.
Yep.
It looks like there are zoanthids in it as well. Let me write this down.
You can start bringing your heading around.
Roger. Westerly, eh?
Yes.
Especially, yeah, just come to starboard.
So, okay, yeah. So just to the right of that, put this in. Just to the right of that sponge, it looked like there was a branching coral, maybe. Right where your lasers are right now. Just go behind those lasers.
Yeah, copy.
If you're referring to that little
Okay,
white
come on
colony, that was a stylasterid hydrocoral.
Now I'm looking at that red one behind the. It's just in the center of the screen now, Scott.
Yeah, there are several branches sticking up, 4 or 5 of them, behind the yellow coral.
Got it. I see it now.
Yeah, I did see this.
Before,
Yeah,
thank you.
I did see the stylasters though. I was wondering about that.
And there's a squat
there's
monster in the
Same
hole.
colony, or the same species that we were looking at that was tucked down by that rock earlier.
Okay. You can see the polyps extended really beautifully there too.
Alright, all set, Watchley?
Yes, we are. Thank you.
Nav, it still looks like 250 roughly to the dive end. And
250 to the dive end waypoint, but directly up the slope I think would be more like 270. I'm not sure how strict that waypoint is supposed to be.
So we have about a little less than 20 minutes left on our dive. I just wanted to remind the science team that as soon as the ROV leaves bottom, we will immediately move into our dive planning meeting for tomorrow. So I know that it's quite late for those of you who are in the Central Time Zone and the Eastern Time Zone. So just wanted to get the dive planning meeting on your radar, and we'll do that as soon as the ROV leaves the bottom. We'll be discussing plans for Chamorro Reef, seamount tomorrow,
And Ifuku.
and Ifuku the
day
next day,
following.
the day following.
Shirley, where are we right now with respect to that wall? Did we go over the edge? Is this,
We did.
on the edge of it?
Yeah, we're over the wall, and we went down about 30 meters, and we're now returning. We're facing to the northwest, and we're going up the slope. A lot of the boulders that you see here have, I suspect, fallen off the scarp that makes that arcuate concave wall that faces to the southeast.